Okay, so next we're going to be hearing from Valentina Loconte, who is the principal Beamline scientist at B24 at Diamond. And Valentina will be providing the B24 user update.
Good morning, everybody. Thank you very much for joining us for this meeting today. I'm gonna give you some brief updates on B24 Beamline. We are basically correlating light and X-ray tomography on our Beamline to explore the cell at the mesoscale. I'm going to give a brief introduction to those who are not familiar with the Beamline, and I'm going to provide some background on the reasoning why we're using X-ray to explore samples using soft X-ray, and then I will provide some outline of the instrument and some updates that have been taking place during the last financial year. So on B24, we use soft X-ray to explore the general structure of the cell at the mesoscale. We don't do this only to identify cell packing and organelle organization, but we trying to do this with the goal of exploring the physiological activity of the cell and trying to understand why the dynamic motion of the cells are actually taking place in a certain established pattern. This can give information about the metabolic state of the cell, but at the same time also to the response to different stimuli that are applied to the cell itself. We trying to do this across multiple different samples of cells, starting from bacteria all the way to mammalian cells. And we decided to do this using a soft X-ray tomography because soft X-ray tomography have some particular features that allow us to look at the integrity of the cells within a thickness that is around 10 micron. Therefore, we can explore the cells in a fully hydrated state, which is representative of the cell at its near native state. This also gives us information to the cell in a fully hydrated state, which means that we don't alter the cell by fixing it or staining it or dehydrating it. We take advantage of the inherent organic contrast that is coming from the organic and bioorganic material of the cell, to explore the morphology and to assess difference in the structure. Specifically, this means that we are working at a specific wavelength, which is around 520 electron volt, which is located in a particular window of the soft X-ray electromagnetic pattern. This is called water window. It's called water window because the water is practically completely transparent to the beam. What does it mean? It means that we can actually take advantage of the features coming from carbon and nitrogen atoms, which are the main component of organic and bioorganic material. So we don't need to stain the cells to track the presence of bioorganic material within the cell. This response that we get from the absorption signal is also directly proportional to the thickness of the sample and to the concentration of carbon and nitrogen atoms. So we can also track a difference in the density within organelles and within the cell itself. Therefore, we can use these features to identify different metabolic state in the cell, or we can explore a different physiology of the cell itself. We use a full field microscope on X-ray microscope on the Beamline. This is an example of how our microscope is looking like, and an example of the data that the microscope can produce. So as you can see, we have a reconstructed tomograms that shows different gray values within the stack. and which can provide information about the different organelles. So, given the different absorption of each organelles and their composition, we basically have different patterns and different morphology that can be identified, which are specific for each organelle. So, for example, mitochondria, as you can see in the movie. I hope you can see my cursor here. Yeah. Mitochondria, as you can see in the movie, are fairly elongated and they are crawling across the entire cell. We can also identify membranes and vesicles related to these membranes. But also you can see from the snapshot on the right of the slide, we can also identify endoplasmic reticulum, lipid droplets, and cell junction, as well as the nucleus. We work at a resolution of 25 nanometers, and this is the highest resolution that we can achieve, but we can also work at lower resolutions, such as 40 nanometers, and this will provide two different types of field of view. At the moment, with the 25 nanometer resolution, we provide a 10 by 10 field of view for each tomogram. We also operate in 3 different modalities of acquisition. Prior to the collection of X-ray tomography, we can collect visible light microscopy images to explore the integrity of the grid and have an idea of the distribution of the grid itself. After we have an atlas, we can select areas that are of interest for our analysis, and within those areas, we can select a region of interest where to collect our tomographic data. So we provide projection in the first place, and then through an automatic pipeline of reconstruction, we also provide the reconstructed stack to users directly on the fly on the Beamline. In general, the experimental workflow does not only include the acquisition of the data on the Beamline, but also includes the sample preparation, which is schematically summarized in here. So we provide support from preparing grids by seeding the cells on the grids all the way until plunge freezing. But also we provide support in exploring the distribution of the cells in the grids, and especially trying to understand the position of some region of interest beforehand, before going on the X-ray microscope. And we can do this by staining the cells with some specific dyes for some targets that can be informative to the users, and this can be explored either with a cryo wide field microscope, but also we have a cryo-sim microscope that can be directly correlatable with the X-ray signal. After we provided the Z-stack of the tomograms reconstructed to the users, we can also move forward with providing some advice about the interpretation of the data and the reconstruction of the data themselves. As I mentioned before, we also have another microscope offline, which is a cryostructure illuminated microscope, which provides a resolution, quite high resolution, comparable with the soft X-ray resolution, the resolution that we get from the soft X-ray data, which is usually around hundreds of nanometers. This allows us to explore tens of nanometer thick cells, and also it allows us to work in a cryopreserved state, so we don't need to work on cells prior vitrification. So this allows us also to understand the quality of the vitrification of the grid itself. The information coming from both microscopes can be correlated. In the first place, we can do this using a software that has been developed on the Beamline and on the fly before starting the X-ray data acquisition, and this can bring to understand the position of the area of interest in the cryo-sim directly on the soft X-ray microscopes. Afterwards, we can provide support to users to perform the correlation between soft X-ray and the fluorescence data coming from the cryosynth microscope. And coming from the cryosynth microscope, correlating the location of specific organelles with the fluorescent signal that has been provided to that has been applied to the organelle itself. And finally, the identification of some organelles is also informative for identifying and reconstructing the structure of a specific portion of a cell, or is useful to identify a pattern within the cell by making some quantitative consideration. Like, for example, evaluating the volume of the organelle or the width, the branching, but also the intensity of the organelle themselves to see if there is any difference across different population of cells. Lately, we have implemented our pipeline on the cryosynth microscope by upgrading our, replacing our processing tool called Softwork with a bespoke on-the-fly processing software called PySim Recon. And Ian Dobby, Tom Fish, and Peter Chang have all contributed in making this possible. So now we have a tool that is available for users that provides direct information about the quality of the data that have been acquired on the cryosynth on spot. So we can basically understand whether or not the area of interest for the users is the one where they are operating, or additional acquisition will be needed. This provides a similar information to what Softwork was providing. We made some evaluation between the two software, and we didn't see any difference. Actually, PySim Recon seemed to be as accurate as what we have before. Also, lately, many more users are being using our workflow to identify the structure of organelles that are not easy to identify within the water window, so they are not carbon-based, for example. And we are going to hear more about this specific example from Alfred later on. Many users have used the Beamline to identify the position of iron-rich vesicles within multiple different cell types. So we can, at this stage, we can map not only the carbon distribution within the cell, but also some specific elements in the cell itself. And for example, in the specific example, and I hope I'm not spoiling the talk for Alfred, we managed to actually Archana and Maria Archilacti before me managed to identify the position of some vesicles enriched in iron in magnetospirale bacteria. So to wrap up our capabilities at the Beamline, I would just leave you with a number of points that could be that can be useful for you to identify whether or not our Beamline could be of help in the future for any one of you. So B24 can help to have a phenotypical characterization of the cell. It provides the capabilities of looking at the cells and their integrity within 5 micron thickness in their native state. With a resolution of about 25 nanometers. It's a quite high throughput, and since it provides the possibility of collect hundreds of cells within a single data acquisition with a relatively low exposure time, which is like 0.5 seconds usually. It is ideal to identify some features or to characterize some cells that are usually non-model organisms, like, for example, tissues or environmental organisms. On top of like regular cell lines, and also it helps to characterize the subcellular structures that have never been seen before, since it does not require any staining or any pre-identification of the compartments, and also allows it's pretty flexible as a tool, so allows it to create a multidisciplinary connection with potentially other technologies, and that can provide more comprehensive view of the whole cell. So this is our team. As compared to EVIC, we are pretty small. We are only 4, but we are going to be here for the entire meeting. Feel free to ask questions to all 4 of us. Archana and Kali are going to be on site for the entire meeting, and Archana has also a poster to show. So please feel free to contact us at any point. Thank you.
Thank you for that, Valentina. Okay, do we have any questions in the room? I can see. I think I can see any hands. Please also ask questions online in the Zoom, people who have any online. Just checking you haven't missed any hands. As Valentina said, yeah, you'll be able to come and speak to B24 team at their stand. So please do ask any questions then if you can't think of anything now. Okay, fantastic. Let's thank Valentina again then. Okay, so that was our facility updates. So now we're going to move on to something a bit different. We're going to be hearing from one of our users, a user of B24, about some of the science that's come out from the Beamline. So we'll be hearing from Alfred Fernandez Castain, and he's going to be speaking about his work on using correlative microscopy completed at B24 to study magnetotatic bacteria.